without further ado, I'll hand over to Bob Kapatic, who's going to be chairing this session for the next 1.5 hours. Bob, over to you.
Very good. I'm hoping it's bouncing up, and I don't see it yet. So let me try again. And normally I'm dealing with multiple screens. Well, I'm really glad that we are together because this is a diverse group of expertise as well as knowledge when it comes to how pulse oximetry can be made better. Now, Sandy Weininger, who you heard from briefly, and I have been involved in this work of standards for nearly 35 years. So it's been a little while. We started with an organization called the ASTM, and this is the same company or group that certifies corrugated boxes, slurries of asphalt on the street, concrete in buildings. And they also had responsibility for medical devices for a period. So that was ASTM, and you'll find the earliest 9919 standard was ASTM based. So we have tried to make sure that the two criteria that Sandy has mentioned, essential performance and safety, are integrated well into that standard. Again, recognize this is a standard for the manufacturer of a device, not a user. There are no clinical guidelines in there. We are actually restricted from the practice of medicine, even though we have clinicians heavily involved as experts. We're not to have that information in the standard. So very helpful to have this gathering because I'm hearing loud and clear that clinicians need better appreciation of the technology, the shortcomings, as well as proper use cases. So in part, that's incumbent on the manufacturers as well, because they need to make sure they're labeling, the official term for regulatory paperwork that's there to emphasize how to use a product, is understandable and as well not in a place that is only residing with the operating manual, so that which can never be found at the bedside. So that said, I'm going to just kind of briefly go over the general topic, this theme of shortcomings, but also the strengths of this controlled laboratory setting condition. So I'm going to take you to this example, which I thought was very nicely summarized by Ellis Monk when he said, look beyond the binary. Well, we were all born into this world, and when doing so, we were all given a binary APGAR score. You were either 0 or 1 on the color scale. Didn't matter what your pigmentation was. It all related to this issue. Is there circumoral cyanosis? And is it present peripherally as well? That's a pretty binary kind of measurement. That APGAR score, as flawed as it is, every one of you has a card somewhere that says your APGAR score was X, as meaningless as it may have been. All right, so this is a very meaningful data point for me when it comes to doing desaturation studies. This is Phil Bigler here. I'll try to circle him a little better right there. And this is Dr. Severinghouse, and they ran out of medical students on that day, so that's me on the table. Dr. Severinghouse managed his hypoxia study slightly differently than where it's being done now. He drove to very low saturation. To be honest, I had difficulty holding my bladder sphincter when I was down at 45%. So this is a challenging. This is a challenging condition, but this is something that is required for manufacturers to meet. And I wanted to make sure you understood that this, especially for those that have not been down that path, there's a reason we do this. And human volunteers, especially ones that are skilled at being hypoxic, become important. And this sort of testing has been done worldwide. This is an example of Hartmut-Gehring, and this occurred in Lübeck, Germany. And this is a desaturation setup. So you see something that maybe you haven't understood or appreciated before, but there's a variety of instruments right there to make sure that the subject is safe. So heart rate, blood pressure, those sort of variables are there as vital signs so you don't harm the subject. But more importantly, you're there to collect data. So there's lots of cables going to multiple pulse oximeters, and you can see the displays here. And that plateau event is what you see there on that old CRT monitor. And in case you wondered if this subject has a means of communication, in Hartmut's lab there was. It was closed loop. Everyone was only looking at the monitor, and if this subject hit that monitor real hard with their foot, they knew the subject was done. So that was the parachute condition. Now, there are multiple sites in which these kind of tests can now be done. And I mentioned the fact that Hartmut is no longer doing that, but that was the case there. And I'm aware of some labs in China that are doing this as well, but I don't have the details on them. But these are the places that you can go to have your device tested. So the slides are available. You can get those details. But the more important thing is, why is it that you need this kind of setting? You need this sort of setting because temperature needs to be controlled. You need to make sure the patient is, or the subject is safe. You need to have an observer gathering data, and you have an observer actually collecting blood specimens, which may be run on multiple co-oximeters. The subject must be in a stable position throughout all of the testing. And you have a variety of gas mixtures that are breathed. And we heard a bit about that earlier. And then multiple oximeters that are in place to make sure that the data are both correlated with a reference pulse oximeter as well as the oximeters under test. Now, there's a large number of variables that can affect the precision of getting the best comparative data possible. Remember that if we're looking to reduce the signal that may be aberrant, and that can come from lots of different sources. So even things like consistent lighting can affect the performance of these devices. So that environment needs to be well contained. And so that is totally different from then going into a real clinical setting where there are many demons or devils that are trying to reduce that signal to noise condition. And that includes multiple clinicians, each handling the blood in a different way, a environment in which you have many, many different temperature conditions or position of the patient that can be changing. And so ventilation and perfusion mismatching can be present. So these are a list of tableized issues that are known to present real world conditions where it too should be optimized. And thank goodness for Equiox and other studies that have taken into consideration those kind of challenges. Now, there are a variety of actual standards beyond the ISO standard, but they're referenced within ISO about making sure that you're doing this sort of thing correctly. So even if you're doing convenience sampling, the device has already collected enough information for submission of accuracy, But FDA or other regulatory bodies are saying, give us some clinical evidence that this thing is appropriate in a use case for a particular population. This is true especially for neonates or children, where you can't ethically desaturate them. So making certain that you're aware of how to appropriately collect the blood. CLSI has lots of standards on that. So just wanted to make sure that you knew that these extra resources are there. Now, I'll give you an example of real world. This is a study in which 11 authors from 6 different NICUs collected bloods. And this is the kind of spread that they had with 4 different pulse oximeters. And this was true over a broad range of values, 92 to 97%. They came to the conclusion that you can have an SaO2 of coming from a blood analyzer of 80%, when in reality it might be 90%. You can be that far off between clinical practice, and I'm not saying it's uncareful or random type of clinical practice. These are situations that are real clinicians collecting blood in a stable infant and putting it in a careful way in the analyzer. So can we assume all the devil conditions is in the co-oximeter? No, it's in largely human practice and perhaps trying to idealize the patient condition by giving them the best saturation they had or waiting for that and then getting the blood specimen. So the real issue that I think is there is, can we better understand the technology so that we are careful to make sure that we are creating the best environment for that testing to occur. So if it's real world, you welcome all of the baggage that comes with that, because that's where the rubber meets the road of clinical care. And in reality, some places really rely on these devices, and so much so that they qualify individuals for when they get oxygen, when they can go home. A baby being released from a NICU has to be checked off in their car seat with a pulse oximeter before they can go home. That's an AAP standard. And we also have this for screening for congenital heart defects. But there's plenty of other examples of what happens in an emergency department and whether or not a person's going to get a nasal cannula. So those kind of clinical guidelines we don't have control for, but we should be able to educate the clinicians that are writing such guidelines to warn them of the shortcomings of the technology. All right, with that in the way of an introduction, I want to turn this over to Paul, who has been running a lab for now. You're going to have to give a history date on that, because Paul, I knew it before you were the one heading it. So it goes back a long time ago.
Right, back 1990.
There you go. So I'm going to stop sharing, and Paul, if you want to introduce yourself, and especially as the baton is being handed off.
All right, yeah, I'm Paul Batchelder, clinical officer of Clinimark, and my disclosure is that I am going to be retiring from Clinimark at the end of this month. So my main goal is to make sure we do the best thing. I don't have any other disclosures. So I want to start with what we have going on here. We're looking at the controlled clinical laboratory hypoxia trial. And that's what we're dealing with. The purpose, as described in the ISO standard, is to test basic safety and essential performance. The FDA guidance refers directly to the ISO standard, but also looking at basic safety and essential performance of the Pulse Oximeter. And so the purpose of this testing in the clinical laboratory is to demonstrate the essential performance, or another way of putting it is the maximum accuracy capability by isolating the device from confounding conditions. And I'm referring back to what Sandy said earlier today, where he was talking about we're trying to remove all performance issues except for how well the oximeter is able to calculate a saturation in comparison to the hypoxic level. So we're looking at the fundamental principles of pulse oximetry design and engineering. We're assuring that they have been correctly implemented. So the goals and outcomes from this test is the best case accuracy capability of the device, which is the essential performance of the pulse oximeter. Additional information that we get is we find dropout. We look at stability. We can look at performance related to gender, performance related to sensor site thickness in the transmission devices, and in some cases, influence of pigmentation. This is not a complete list, but that gives an idea of some of the things that we do see with these in this type of test. Now, we control for in the clinical laboratory, as Bob so carefully put it, normal perfusion, no movement, correct sensor placement, sensor crosstalk, and normal levels of hemoglobin. There we try to control for the time sampling differences of the reference co-oximeter draw site and the device under test. We do our best to look at the blood flow lag time between reference and device under test. So there are challenges to implementing a multifactorial design in this, and maybe John Feiner can talk a little bit more detail about this if he has time. But basically, multifactorial design enables you to put many different parameters in or influencers into a study. And you can tease out the various influences. So motion is an example of this. So we do motion studies, and with the you can combine it with an accuracy hypoxia trial at the same time. But they have to be different hands. You really, or at least you have to pause and let things settle out before you can actually look at the actual best case accuracy of the device if you're doing a motion test. Low perfusion is probably an area, for example, that we cannot combine with the best case accuracy performance of the device because of the perturbation that is impacting the SpO2 calculation. So we're really kind of restricted to only testing best case accuracy and removing all of the other influences. Now, something that's very important in this type of test is are the pre-analytical and analytical techniques that were referred to a couple of times earlier today. Pre-analytical sample preparation, air in the sample, sample mixing, etc., is very important. And then the analytical is minimizing the measurement error with the calibrations and quality controls and increased vigilance in real-time related maintenance, like cleaning, clearing plots and film during data collection or running a sample in the co-oximeter. And in the clinical environment, it's not as amenable to spending all this extra time with the reference co-oximeter device. Now, an interesting piece here is that the reference equipment accuracy, the co-oximeter, its accuracy is 1%. It's what's published in the two most widely used co-oximeters, which is instrumentation laboratory and radiometers. And when one considers that the published accuracy specs of pulse oximeters are 2 or 3%, then the inaccuracy of the co-oximeters are of great importance. There's no standard reference material for oxygen saturation, so who knows what the co-oximeters are really measuring. The co-oximeters that we have today are previous reference compared to past devices. So the ground truth that, for example, with the band slight method, it's very time consuming and it's technique dependent, very complex. Well, that hasn't been done in any of the co-oximeters that are on the market today. They've all been reference compared to recent past co-oximeters. Now, there is a difference in co-oximeter calibration curves. The co-oximeters are used for the calibration reference, and most pulse oximeters were calibrated to either an IL, instrumentation laboratory co-oximeter, or a radiometer co-oximeter. In the last few years, some companies have used an average of data from both from the two of the co-oximeters. Different costs, different cals. The issues at the calibration curve of the two brands is different. Yagi published a 1.5% difference between IL and radiometer. Gehring published a 1% difference, and Clinimark data shows approximately 0.5% difference or bias between the two widely used co-oximeters. What this means is that this was what we've done in the past is about half of the pulse oximeters were calibrated to the radiometer, while the other half were calibrated to the instrumentation laboratory co-oximeter. This can cause quite an issue. So this is an example of a study that we conducted, and this was actually published in 2007. It came with the IMPOV conference at Duke. We have 3 instrumentation laboratory co-oximeters and 3 OSM radiometer co-oximeters. And we ran multiple samples, each one in every co-oximeter 3 times, over a range of 70 to 100%. And what we found was that there's about if you average the IL, which is more noisy, by the way, than the radiometers. That's about 0.2% of radiometers down to minus 0.3%. So it's a 0.5% off between the two primary devices. What that means is if you have a pulse oximeter that was calibrated to an IL and you test it in comparison to an OSM3, you already may have a 0.5% inaccuracy built in. So taking this bias into consideration, it's obvious that your pulse oximeter could fail if you use the wrong reference device. Now, an improvement to this is to use multiple co-oximeters, and I know that both the UCSF and Clinimark do have multiple co-oximeters, and perhaps both co-oximeter calibrations could be used. This is something that we haven't discussed in the committees, and it's something that we should at least entertain improving. So using multiple co-oximeters, you can decrease the error of precision, and if you have both calibration curves, you can also correct for the difference between the two co-oximeters. Now, sampling stability methods is what I'm going to end with here. I'm looking forward to John Weiner's presentation. He's got a much cooler presentation than I do on this. But the purpose is to maintain a stable such SaO2 period, so that while we're drawing the arterial sample, here's an example of 4 samples or 5 samples being drawn at each of these plateaus. And you'll see that we have a little bit of drift intentionally in this graph because that's kind of reflects reality. But what we need is a stable sample period at the period of time during the period of time, we draw the arterial sample. And we need to then compare that to the pulse oximeter. So not only do we need the SaO2 to be stable, but we need the SaO2 at the blood draw site to be the same SaO2 that is at the sensor site. And so there is a difference in that. The timing is of utmost importance. UCSF's saturation stability method, and I stand to be corrected on some of these, but the participant is asked to maintain a relatively constant respiratory rate by following a metronome at times, I believe. The purpose is to maintain steady ventilation and to avoid deep or shallow breathing that could result in instability during the sampling period. The projected SaO2 is calculated from inhaled and exhaled gases and displayed in real time. And also, the lab view is used for establishing the stability here and incorporates both the calculated saturation and the reference oximeter readings. And it gives kind of a red light, green light to go or no go for a stability period for a sample. And inputs of P50, 8A gradients for CO2 and O2, and base excess is used to calculate. And the carbon dioxide is administered when necessary to maintain normocarbic arterial values. And then, like I mentioned earlier, 2 clear pulse oximeters are also applied. This is a rough picture of the calculations that's made. And this is a picture of an old picture of the laboratory at UCSF. That's Dr. Severinghouse. I'd like to highlight, and I think that he probably should have received the Nobel Prize, in my opinion, for the work that he did on blood gas analyzers and the CO2 electrode and so forth. I don't know who this gentleman is back here.
Can you blow it up?
Yeah, you can.
Back with hair. I can't tell.
So,
Yeah, I can't tell for sure, but I think that's you.
Yeah, it moved to your chin, John.
There, and then someone stole it under this. I didn't just lose this. This was stolen.
So at the Clinimark labs, we have a different approach. So all participants are evaluated in a pretrial hypoxia trial in order to characterize their physiological profile of the response to hypoxia. And this is not in conjunction with any device under test. These types of tests are done in our laboratory before we do any use them for hypoxia test trials. And the reason we do this is that over the years, we have found that similar to the blue bloater and the pink puffer in COPDs, where the blue bloater in the COPD is comfortable, his hypoxic response or his response to hypoxia is blunted. He doesn't feel as uncomfortable with a low saturation, and so the person breathes more relaxed. They're hypoxic because their lungs are crap, and they're blue. Cyanosis from that in many cases. The other end of the spectrum is the pink puffer. The COPD patients whose lungs are also equally as bad, but they have a very high level of discomfort with hypoxia when hypoxia is present. So they breathe, they puff, they breathe harder and maybe deeper with larger tidal volume. Even though their lungs are crappy, they're able to keep their saturation up, and so they maintain a pink condition. Well, we found that in the population at large has the similar response, but their lungs are fine. So with some individuals are very comfortable and have very stable SaO2s, SpO2s at lower saturations, and others are just very uncomfortable and in a small amount of maybe about 5 or 10%. They just keep changing their tidal volume, and at some point they can't maintain a high saturation. And if we're pulling down the FiO2 gradually during this whole time period, and they're still not moving off of 90, suddenly they'll plummet and become unstable. So we look for individuals that are generally moderately or very well accommodated to a lower hypoxia. The participant during the study is coached to maintain a comfortable respiratory pattern and to a degree is allowed to set their own pattern based on their own physiological response to the hypoxia. ETCO2, end-tidal CO2 is continuously monitored and feedback is given to the participant to adjust their tidal volumes in order to maintain a normal tidal CO2. And carbon dioxide may be administered if necessary to remain non-toxic. Transfer standard pulse oximeter is utilized to identify stable plateaus for blood draws, and then we use a forehead sensor as a weather vane to identify sudden changes in saturation. Picture of these are also old pictures of the Clinimark lab, and this we had some 682 ILs, and then we had the OSM 80s all being run at the same time during the study, and all the samples kept run in all devices. And then an old picture of the of hypoxia. You can see here we have a mask and flow by, so it's open system as well, and we changed the flow rate of the gases going through past the subject to match their, or actually overwhelm their tidal volume. And then we plot the data real time. The inspired gases are controlled by the computer, the operator here. This happens to be Dina Rayleigh, started designing pulse oximetry sensors in 1982. So go figure, you can do the math. And then the information is displayed at real time, transfer standard regressed, so that we can see what's going on with the pulse oximeters as we're going along and see what kind of stability we have. But let me make it clear that we only assess stability based on the controlled pulse oximeter and not the device of the test. So that's my story, and I'm sticking to it. I'll turn it over.
Where are we going?
And John, from your perspective, do you, I've certainly known you for a long time as a,
Oh,
oh, there you are. We have echo.
Okay. There you go. I wasn't sure what I was supposed to be.
So John Feiner is a practicing anesthesiologist here at UCSF. I've known John for, my goodness, probably 30 years, and knew him in many roles, and more recently as the head of the liver transplant anesthetic side. And he's given up that a bit, but he is spending a lot of time still on statistics related to modeling of various conditions. And you saw some of that in Dr. Bickler's presentation, where they were trying to make understandable in a graphic form what can happen in the way of splaying of data with multiple variables. And so that's really John's work. So.
Yeah, I'm retired and failing at it every day. They keep bringing me back in. Dr. Lipnick asked me to show you what we're doing kind of as a QI project, but also gives me an opportunity to show what a desaturation run looks like on a simulator, where I can take a real study that was done and replay it at high speed potentially and see how we're doing. As Paul mentioned, one of the key goals here is the stability of the desaturation. The technique that Severinghausen developed for testing was based on the idea if the plateau is perfectly stable, then any uncertainty between the pulse oximeter and the blood draw goes away. And that's the key thing, the whole issue of timing. Perfect plateau means that timing uncertainty is decreased. We use a calculated saturation, which was based on Severinghausen's formulas. This is not what goes into the final statistics, but allows us to control the levels and particularly distribute the data throughout the saturation range. We do have fudge factors that can go into it that improves it so that it's almost perfect, and it can sometimes match the blood gas data better than the pulse oximeters do. It is also necessary because of the time delay that gives us breath-by-breath control, so we really know exactly where we are. We do use CO2. I am vigorous responder to hypoxia. It makes me a safer breath hold diver, but not as good in some ways. So I tend to, if people get sleepy, they're low response, I tend to have the CO2 go up a little bit more. People really short of breath, you can modify it and make the study much more comfortable. We also use a rebreathing system, which means if someone decides to take a deep sigh, the FiO2 is automatically going down. Even a computer-controlled system would not be able to respond to that. So it helps stabilize the plateau. Again, John Severin helps set up our circuit. I've always been fascinated by projects like this, where I can try to improve things and really leverage the computer for what it's good for. I don't fully believe in AI. I believe computer-assisted humans and getting the computer to assist in a way. So I will just show you what we're dealing with here. This is a plot of, I'll start running it. You can see these are the pulse oximeters. We have two different pulse oximeters here. And I've come up with some parameters here. I thought intuitive, we want a slope of 0, but if I measure the slope, not a very intuitive number because it's going to have many decimal points. So I'm really trying to come up with an algorithm that represents the 60-second change in oxygen saturation. So that's a more intuitive number. Very important to try to come up with parameters that are used for an algorithm. I mean, we would want it to be 0. And I'm going to look at the stability. The first stability is going to be the calculated saturation, and then the pulse oximeters have stabilized. You can see what a real study looks like going at 20-fold speed here. The dark blue line is the calculated saturation. Notice the pulse oximeters came down rapidly, and this is a pretty good plateau. I'm getting green lights on both here. Now the Massimo is a little off. Now this is the new level. So this is what we do. We try to rapidly go down, hold it. There's a little slope here, a little off, going down again. And you can see the pulse oximeter, this is a very warm study subject, so we're getting good rapid responses, and then we resaturate, it comes right back up. Now you can see the heart rate coming down here. This person probably had a pretty vigorous hypoxic response. We have a little rest period, we catch up on blood gases, so we're not going too far behind. And then we can start on this is variability as they're sitting here sometimes. Partially from hyperventilation, we have a second run, nice and stable, heart rate goes back up. This is the CO2 down here, which is nice and stable. This is the O2. We have another run, good response, little instability at first, not unusual. We stabilize, hold it, draw multiple samples, final desaturation into the 70s. And often with the different levels, what we're trying to do is get a few samples in the upper 90s, a few samples in the lower 90s, upper 80s, lower 80s. We do each of those on the different runs, and we can use our oxygen saturated or calculated saturation to be able to do this. I'm trying to come up with some intuitive displays so the corner of your eye can help people. And it takes a while to perfect this algorithm, but I can do this offline. We can do this to evaluate quickly how good a run it is. And ultimately, we would like to reject bad data based on objective criteria and not bring in bias just because it doesn't look good and it doesn't match the blood gas data. We want to try to make this objective, something Mike Bernstein and I have been talking about for some time. And it's been a thought process. Now that I'm retired, maybe I'll have some more time for this if I can get some of this off the table. Now I want to play a different one. I may slow this down here in a second. Here comes this. What I'm going to do is slow this down a little bit and show this is an example of pretty low perfusion and how long it can take for the pulse oximeters to catch up, and what difficulty we have with this. These are just starting to come down. Been here for a while. They're not quite matching here. And this is very, very low perfusion. It's stable now, but you can see a pretty big difference. I don't remember this is a dark subject and a reason for positive bias, but with very, very low and slow circulation, we have to extend the study for a long time. And it can make it a lot more challenging than to run this through. You can see here, again, the desaturation, a little instability at first. We're doing the dials and look at the difference in time for this to come down compared to that first example I showed you. Heart rates are not as high in here. This is probably, the heart rate probably correlates with what Paul was saying about people's breathing response. Look how long it takes for this to come down. So this is a good example of a very, very challenging study with no perfusion where we have to wait a lot longer. Notice the heart rate comes down here immediately because, and look again how long it takes for resaturation, but the heart rate is transduced immediately. So when we give oxygen back, you see the heart rate, people with high heart rates, the heart rate will plummet immediately even while the saturation takes time to catch up. Probably pretty good correlation between the heart rate response and the ventilatory response. And again, this is something we also talked about when we published this whole issue of silent hypoxemia, really relates to the normal variability of responses. And again, so this is just a very good example. Still working on the red line, green line here. We're trying to work this in, particularly as we have some newer people doing this. And I think when we look over, Phil and I have been doing this for many, many years. And I'm trying to, again, turn this into something intuitive. In some ways, it's always harder to do a good run when there's a lot of delays. And again, a little overshoot, but again, we get this immediate thing. So we don't have something on the forehead. We've been doing this for a long time. We don't bring in people to pre-test. We hopefully have taken a chance and get a lot of good study subjects, but we have a lot of people who've done this over and over again, which gets to the question, could hypoxia be addicting? It may actually be the fees. I've told many of our medical students that this may pay more per hour than they'll make if they go into pediatrics. So again, hopefully for the people who have never seen one of these studies, this gives you a sense of what a real desaturation study looks like and the challenges we have. I didn't find a good example, but on the lower saturation levels, we sometimes will see a stable calculated saturation and a drift down of the pulse oximeter values, which probably represents an AA gradient, and I believe it's more likely diffusion limitation, which is something that is well known at altitude, and due to inadequate time to equilibrate when you have low CO2, based both on increased heart rate and increased respiratory. So that's a known issue at altitude. I don't think it represents lung water or anything, but it can be an issue. Both the stability and some gas exchange changes down in the 70s are very, very noticeable. And will always result in probably worse data at the lower saturations, more noise, independent of whether the pulse oximeter is actually performing well. The pulse oximeter probably doesn't perform as well in those areas either. They all have a sweet spot, which is probably why the FDA definitely wants nice even data over the ranges. Hopefully this gives you a flavor of the challenges of this and the noise. This is biological data. It is noisy. It is more challenging. It has errors, and even if the gold standard or perfect gold standard, maybe it's only silver standard, it's still probably much closer to the real value than certainly the pulse oximeter. But there are certain real issues trying to get good human data. And this is in a very controlled setting rather than so having a simulator, I think, will allow this to be much more useful. And certainly developing it allowed to a good display what a real desaturation study looks like for those who haven't witnessed it or those who haven't actually been a study subject. Dr. Bickler and I would trade off many years of putting each other's A lines in. And in fact, I think we never even decided. We flipped a coin once, and it's like, okay, you've got to be the study subject. He said, no, I'll be the study subject. Keep my A line in. We had many issues because Dr. Severinghouse did the gases, but didn't put the A lines in. And in fact, I think you once came down. I was working the OR. You came down. I put the A line in. You went back upstairs. So we've been study subjects. It is a challenge. My lowest saturation is 48%. I remember that you had me beat at 46%. Except I was post call. I had to sleep, so that made it a little bit more of a challenge to feel what it's like at 48%.
And did you manage your bladder?
I did. I had no problem with my
I
bladder.
never went to bed. Okay, don't get me wrong.
I didn't.
The
I had
urgency
trouble
was there.
maintaining consciousness, which probably is lost. Now I think again, you really, even the 50s and 60s is probably safe. It's miserable for a lot of people, but when you're targeting 70% for the FDA studies, you sometimes hit the 60s. So it is essential to realize that we have a pretty good safety margin. That level of hypoxia is better tolerated than people think. And certainly data, even at loss of consciousness, you're not really in danger. The brain is probably shutting off, but then you are at a slippery slope. You don't have that much time before that is bradycardia for any of us who practice clinical anesthesia. And I don't lose airways. I've misplaced a few and had to have help, but I've probably come in to help many more of my colleagues who are losing the airways and seeing that thermal desaturation and how frightening it can be in an OR circumstance. The lowest saturation I've ever seen on a well-working monitor was 9% in a patient, and they did have bradycardia. Again, not my case. I was helping. So always happy to entertain other questions, and I will pass it on to our next contestant.
Well, thank you for your perspective, and I have to say that kind of covers the spectrum. I knew Dr. Bickler first. He's a UCSF or UCSD grad as well, but he was in John West's lab dealing with high altitude physiology, and I was there trying to do oscillation on geese at the time. So that was my undergraduate project. So he did it at high altitude. He still does. They do such studies at altitude at White Mountain, and they put in PA catheters while up there just to make sure what's going on with heart function as well. So recognize that when John then says, this is what I have done for breath hold diving, that's reality, and they have studied many of those kind of divers. And I'll mention as well that David McLeod, listed as one of the resources, that the PI at Duke has studied and published many of those diving conditions where CO2s rise, individuals can acclimate to that, and their O2 levels drop quite low, but it's well tolerated for tens of minutes underwater in breath hold conditions. So
the record,
Let's stay on focus.
okay, yeah, he might. Okay, so I can only refer to what I saw from Greece in the Mediterranean. But all right, so our next presenter is actually did much of her training in New York at Mount Sinai, and a place that I've done a lot of work as well. And she's completed her fellowship here at the Zuckerberg, San Francisco General, and has been dealing with a lot of the health equity situations needed in perioperative management and assessment of patients before they come in for their hospitalization. And she's currently focused on equity as it relates to pulse oximetry. So Boomi, the floor is yours.
Hi, everybody. Wow, I'm in an interesting position following all of these experts today, but thank you guys for having me. So I'm going to talk about something a little bit different. I'm going to give you guys a little bit of a food for thought, challenges to achieving health equity in pulse oximetry. So over the next few minutes, I'm going to reiterate what our goals are here as a consortium, talk about why diversity is central to these goals. I'm going to outline some challenges to achieving diversity, and then talk a little bit about what our research group is doing as a subset of the hypoxia lab at UCSF, and offer some solutions. All right, so let's talk about our best laid plans. For any endeavor, we need to be clear about what our goals are in order to reach them. So What are we doing here as a consortium? As I mentioned, we're here to answer the question, what does health equity in pulse oximetry look like, and why is diversity important? Specifically, we wish to elucidate the role of skin color in pulse oximeter function with more studies. We want to catalog device data and make it freely available so that patients, healthcare facilities, and even research facilities can make more informed decisions. We want to overall create better devices that work well and equally well, and ideally for all patient populations. And then all those things together would help us to improve equity in patient outcomes. So I know that a lot of us are probably familiar with this quote, even if we don't remember reading this book in high school. It's from Of Mice and Men, and that is, the best laid plans of mice and men often go awry. So let's talk a little bit about intention and reality. So chewing over what we're doing here, the role of skin color in device function, as we mentioned multiple times, has been a topic of debate, and therefore it must be clearly defined. So skin color alone, as we know, may not be the only variable leading to observed disparities in pulse oximeter function. And we've learned that most likely other variables such as perfusion, when combined with skin color, may be playing a larger role. However, we first have to do a better job of isolating skin color as an independent variable and measure its effect. Therefore, methods of categorizing skin color must be updated. We know that using subjective measurements such as the Fitzpatrick scale, we haven't done a great job of this, but there are plenty of good folks here who are working to tackle this issue with new methods and new scales. In addition, we must be intentional about representation in clinical studies. What I mean by that is we need to put in the work to diversify our studies, to include more people, and particularly, as is our subject here, people with darker skin. So to achieve these goals, we'll need to review and improve our recruitment materials and practices, as well as endeavor to include underrepresented populations, and this is all very important. So we need to be aware of pitfalls as we move forward through this endeavor. So an important fact to acknowledge is that underrepresentation of racial and ethnic minorities in clinical trials remains a reality while they have disproportionately higher rates of health disparities. In addition, failure to adequately represent populations in clinical studies threatens the generalizability of results of studies. So that being said, we have to be mindful of a few things, not the least of which is our tendency to oversimplify and conflate skin color with race and ethnicity. Things are not black and white, as they say. Dr. Ehi gave us beautiful and succinct definitions to differentiate between these terms. And Dr. Monk expertly pointed out that keeping in mind that there are deeper levels of nuance and diversity, even within diverse categories of underrepresented populations that we'll be working with, is an important, keeping in mind is an important step towards preventing unintentional harm in these communities. So let's talk about barriers to recruitment in studies, and particularly barriers to recruitment in studies of healthy participants. So we should acknowledge first that working with underrepresented communities requires us to think critically about the complex social and historical barriers to enrollment, and to make attempts to disrupt these barriers where we can. But some of the barriers are very easy to understand and maybe less challenging to address. And one of those barriers is awareness of the studies. So the list that's going to follow is a non-exhaustive list. These issues that I'm going to list came up various times in my research, including in studies that are as recent as 2019 and 2021. And the first of which is awareness of the studies. So obviously, if we're going to be doing a study, we want to make sure that we're putting the word out there that we're having the study done. And this is something that's easy to address. So where are potential volunteers hearing about the studies? How do we get the word out that studies are occurring? Where are we putting our flyers and our recruitment materials? And then next is access to studies. Will potential volunteers have difficulty coming in for studies? Are there scheduling issues, distance, travel, cost, disability issues, all of which are factors that need to be addressed? And then next we have communication barriers. Are there purposes, are the purposes and goals of our studies being made clear? Are there culturally appropriate recruitment materials available? And are different languages being offered? Next is the perception of benefit. So what is the incentive for a healthy volunteer? In my reading, I was coming across many clinical trials that were intended for patients with chronic illness or cancer, all clinical trials that offered a potential treatment. So this is clearly not a benefit for a healthy volunteer. Also, is the study going to be time consuming? Why would someone agree to give their time to the study? How do we address how to make the study worthwhile for a healthy participant? And then it's very important for us to always mention the historical context and potential mistrust. So while people may not have personal prior negative experience, there may be mistrust within the community secondary to the historical context. The examples that come up repeatedly, of course, are the Tuskegee experiments and experiments that took place within the field of OBGYN. And then related to mistrust, some people are simply concerned for their own privacy and may be reluctant to participate. They want to know exactly what's being done with their data and personal information. So keeping in mind these barriers, a thoughtful approach can increase enrollment in studies while fostering relationships with the communities that our participants are coming from. So I just wanted to share a little bit of light. I know we're talking about barriers, but there are also reasons that participants cite for wanting to be included in studies. So the ones that I'm about to share are from a 2019 study that was published in the Frontiers in Cell Development and Biology journal about why a focus group of participants chose to take part in biobanking studies for genetic diseases. So the first one was pure altruism, which is great. They wanted no personal gain from the study and just wanted to do some good, which is great, probably rare, but is great. Next is a personal desire to be part of something big and positive. Can't fault anyone for that. A desire to find solutions for themselves or for their families for illnesses. And then finally, to help improve treatments for future generations, particularly grandchildren. Okay, so let's talk a little bit about engaging communities. So addressing barriers to enrollment in certain communities necessitates interventions at multiple levels, both at the community level, participant level, and provider level. So how do we do this? How do we improve recruitment of underrepresented groups? One possible solution would be to advertise and recruit actively. What I mean by this is to address issues of awareness and access. We want to take a good hard look at the recruitment practices of our labs, what is being used. Are they outdated? Are they culturally sensitive? Where are they being placed and how are they being disseminated? And then again, what media are being used to spread information? If we're using social media, do people have access to computers? If we're advertising on TV, things like that. The next step potentially could be to diversify your team. So this one goes a long way. Potential recruits for studies will benefit a lot from a study team that makes them feel comfortable and, you guessed it, maybe even looks like them. So taking that into consideration when building out your teams for studies and hiring is very important. Also important is involving community leadership. So this could be family physicians, health advocates, faith leaders, and community representatives, all who can help to spread information about studies, and also address some issues around mistrust. And then, of course, very important, we want to practice effective ongoing communication, look into offering language services, and where appropriate, other forms of communication, two-way communication. So feedback surveys from the community, as well as things like newsletters, blogs, and community events to share progress of the trial or research, so that participants can know what's being done with their information. So I am helping to lead an initiative at the UCSF Hypoxia Lab. We've called ourselves the Equity Team, and we are endeavoring to come up with some solutions. So over the next 6 to 12 months, our goal for this team is to work on an important objective of the overall Open Oximetry grant project, which is designing a toolkit to improve diversity in medical research studies. So what we're coming across is that this toolkit is less going to be reinventing the wheel and more bringing resources together and talking about what has worked and what hasn't worked in the past. Another thing that we're doing, as I mentioned before, is building a diverse team. Our team consists of attendings, medical students, residents, engineers, researchers, organizers, research coordinators, all of which we're very fortunate to have. A lot of members of our team are already have a vested interest in solving these issues, and this was achieved through thoughtful recruitment. And then background research and information. So our team is working on reviewing literature, not only about bias in pulse oximetry, but also strategies for attaining equitable research practices. So of course, arming ourselves with knowledge is important, and we encourage our members to come prepared. And then updated recruitment materials and practices. So one of the immediate goals of this group is to improve our admittedly outdated recruitment materials and practices for the Hypoxia Lab. Then what we want to do is share how we specifically accomplish this goal, and that is an ongoing process. But of course, data is everything in the digital age. So to that end, when we put out these new materials, we want to tag them and allow feedback. So this will help us understand engagement with the material that we're putting out. It'll be most likely something as simple as a QR code that can be scanned. It pings how many times the QR code was scanned, and we'll have different versions of our recruitment materials. And in that way, we're collecting data about what is working, what is not, comparing and contrasting. The community feedback survey is another goal for our group. This is to perform an assessment of our community's general knowledge of health disparities, and more specifically, knowledge about hypoxia. So this feedback will help us to tailor our recruitment and education materials, which may help us to increase our enrollment in studies down the line, and will also help us to build that guidelines potentially for other people who are trying to diversify their medical research teams. And then finally, idea sharing and collaboration. Forums such as these are very important for us to have. We're making a conscious effort to continue working in these spaces and bring in collaborators so that we are not siloed and we can bring together all sorts of different ideas. And I think that this forum is a wonderful step towards that goal. So overall, the goal of this team is to lead the way for conducting studies with equitable representation and hopefully even beyond the pulse oximetry space. So proceeding with caution, again, how can we move forward ethically and responsibly? So some important things to note here. So we want to clearly communicate risks and benefits of all studies. I already mentioned potentially offering language services. health screening questionnaires for safety. So one of the things that we talked about is that we are trying to recruit healthy volunteers. We need to balance the recruitment of healthy volunteers with which incentives that we're going to be providing for them. So we don't want to have the incentive be such that someone is trying to join the study who is not a healthy volunteer. So that's one of the things that we have to balance. And then, of course, being well educated about the communities that we're approaching, and being flexible and forgiving with ourselves because we are all learning as we go. Okay, so a quick summary. So even though we are well-intended, we must make sure that we have clear goals, which is always important, and remain diligent to avoid pitfalls, especially unintended harm for the communities that we're working with. We want to address barriers to enrollment, and they can be overcome with deliberate, conscious effort. And then we want us all to be leaders. So we have a unique opportunity here to take an issue that has a lot of attention on it and make some meaningful change and be an example for other issues within the medical field and potentially beyond that are also trying to make meaningful change. And I hope that this collaboration will be a win for health equity. Thank you, guys.
Healthy
Hold on one sec,
volunteers.
Bob. Sorry, now you're off mute. Go for it.
Oh,
You're good to go. Sorry, I had you on mute.
there are a lot of people that would like that. Okay. So the concept of getting someone to get enrolled is a challenge. And I greatly appreciate how there are multiple barriers to doing that. But realize again, when it comes to these desaturation studies, ethics plays a huge role. So there is an ethics committee outside of the United States, and within the US, there's an investigational review board that considers these protocols and signs off with the institution and the PIs accepting the liability associated with making sure these are going to be safely conducted. So those are realities, and therefore at the same time, since they're healthy volunteers, those are naturally barriers to real world data because they don't have comorbidities. There aren't issues associated with other conditions that would put even others at risk, such as you test for pregnancy and you make sure that their carboxy levels are within normal ranges. So they didn't recently smoke and then during the desaturation study end up with changing carboxy levels. So an interesting set of questions that therefore come in. And so I appreciate that. And there's some other active comments that are in there, but I want to make sure that some of that is at least understood and addressed in advance. So one of the issues that came up is note these characteristics of idealized studies designed to have only one parameter, and that is SaO2. And so are there issues when comparing performance between device standards or a potential predicate? So the predicate that is typically used for substantial equivalence is not there under study. That is something that is simply used for a submission, at least with the US submission. And so you're not directly comparing it at the same time in a test subject. Rather, the predicate device has various different features which you wish to match, one of which is making sure not that it matches performance, but that the performance is within the ISO and FDA guidelines. So you're never trying to dead on match a substantial equivalent device. I just want to make sure that that's clear. There was a time when you could, when devices were available, and you can still make them yourself, where you would tonometer a blood specimen to a precise saturation. and you could take that gas tonometered sample. So you would spin a blood sample, create an environment in which the gas was absolutely at a P50, for example, 50% saturation, and you could put that into your co-oximeter. Those devices are no longer available. And you are, as Paul mentioned, reliant on whatever the QC material is from the manufacturer. But that's the reality. And lots of these things have changed with time. Another example is Dr. Severinghouse used rotometers, multiple different flow meters, to create the exact gas concentration that somebody would be breathing. And it was titrated against a mass spectrophotometer. Mass specs are no longer available, but that device, the PerkinElmer, went for many, many years with lots of tweaking to keep it alive. Now there are even better gas analyzers. So if you need those kind of details, they certainly are available. UCSF is using one such system. Duke is using another. I know you have systems, but I don't know if you're matching the UCSF or the Duke one.
I'm sorry, I was trying to respond to a.
Oh, gas analyzers. But they need to be real time. That's a critical.
Yeah, they do. They have to be responding in timely action.
All right, so certainly stresses within the chat that it's functional oxygen saturation that you need to be using as a comparator. So if you're doing real world testing or in this controlled setting, make sure that you're using the correct saturation for comparison. Now, another issue that came up is understand the need for control lab setting studies. Does this method of screening healthy subjects select out a population that has a distinct cardiovascular phenotype that may behave differently than some and much of the general population? You are pre-selecting and even pre-screening to get this group of individuals. And oftentimes, as you heard, they're cycling back through because you know they test well. And in the end, you're talking about some rather basic physiology. Does gas move from the alveolus into the pulmonary capillary bed? Does it get well enough brought in to the left ventricle such that you have systemic circulation that will take it to the finger in a quick enough fashion to give you the data and the stability in that data? So inherently, there may be individuals with unknown shunts, both pulmonary or vascular, that you are weeding out. That's not the general population, therefore. But at the same time, well people, unless they're going in for an elective procedure, are not the ones that need the pulse oximeter in the emergency department.
Can I chime in just for a second? I think the question there is, this is Sandy Weininger, FDA, whether the physiology is different for sick individuals, such that the oximeter isn't going to be able to properly detect the number of oxygen molecules bound. So if you can postulate that during disease that changes, then sick individuals then oximeters calibrated on healthy individuals may misrepresent saturations on sick individuals. But if we can't postulate that there's a cause there, then, so far, at least for the last several decades, we've assumed that oximeters, pretty much are measuring the oxygen saturation.
But Sandy, isn't perfusion an example that argues kind of the point that you're saying? I mean, isn't that enough? Isn't that adequate? We know perfusion causes, we see routinely as clinicians, problems in the accuracy of the pulse oximeters in patients we attribute it or associate it with. It doesn't prove causality. with low perfusion, but low perfusion is not part of the, the scenarios that could be accounted for even in healthy subjects, but is not currently.
Well, I think Mike is still scratching his head to figure out why low perfusion causes the types of bias we see. He may have already scratched his head and has the answer, but I think that remains to be seen, to be proven. Certainly, we need to incorporate low perfusion studies to see how the devices respond, and it would be really nice if we could figure out what low perfusion actually is and what's causing the degradation, but I don't think we're there yet.
And we'll certainly have a separate session on perfusion related issues. Leo, and then Phil.
I guess this brings up a kind of a question I had asked Phil earlier. If you think about it, we have SaO2, our ABG, as our gold standard lookup for SpO2. What is the gold standard lookup for perfusion index? Do we have a measurement that we can use? Because when we look at perfusion index from a pulse oximeter, it is a value that is derived from the pulse oximeter itself. So if we're worried that the pulse oximeter is not accurate, we're kind of like a chicken chasing its tail, or whatever the term is. I'm not sure what we're talking about, but it's like we're trying to pull ourselves up from our bootstraps, right? We have a device that we are concerned about its accuracy, and then we are using an output from that device, the perfusion index, as a marker of accuracy. So what is the gold standard that we can use that's the analogy, that's analogous to SaO2 in an ABG, that we can say, in fact, is there an ISO standard that says perfusion index must be accurate to some level? How do you measure that? How do you even assess whether it's
benchmarking? That is problematic, and regrettably, and it's true. There's IP around some of that,
right?
So people aren't necessarily willing to share.
It very well may be that perfusion index is the surrogate for the same issue of skin tone that we're talking about.
There's certainly an argument for that, and Mike, I know that there was a move afoot to have PI be a breadboard condition that you will have open source.
Yeah, we built something, and Leon and I were talking about him making some more. The idea is if we record the raw waveform data, we can process it like manually, make sure we're doing it right, and figure out the PI.
Yeah.
And going back to another thing that you said about the hemoximeter, the newer radiometer hemoximeters, it's much harder to keep track of and determine if they're working. We had an experience with one that had a hemolyzer problem that we didn't know about. It was probably a hemolyzer problem, but it stopped working and there was no external indication that it was drifting off.
Yeah.
But see that point about adding it on the chip, that's still using the pulse oximeter to get it. Is there an independent way that we can measure perfusion index? What is that physiological variable that we can use as a ground truth against which pulse?
Well, it's
So
local physics. I only am bringing up Mike because I knew that I was hopeful that this open, condition would be available. I want to get to Phil first because I think we beat that one, and more importantly, we're dealing with perfusion a lot tomorrow.
Well, my comment is going to relate to the applicability of the lab studies to the real world. We clearly need more real world studies to see whether the lab environment really is recapitulating clinical scenarios that we see patients. That's essential. I mean, the FDA is funding that. We're still a ways away from having answers. I mean, solid real world clinical studies of pulse oximeter performance have not been done. Before the FDA took the initiative to fund this, all the studies comparing pulse oximeter readings to some reference standard were flawed in various ways. Either the saturation range was too high, or enough low saturations, the timing was off. We're basically flying blind until we complete studies like Carolyn's. We're not going to really know in what direction to go with the lab testing. right now, we're behaving like scientists. We're controlling everything. We have optimal conditions. So at least we can say that in the lab, there aren't known inconsistencies that will bias our data. How that data applies to real patients in real environments remains an unknown.
Indeed.
May I chime in? What we don't have yet that we have in many other ergonomic situations is the range, the dynamic range and the spans of the parameters of interest. You know, so if you're going to go design a car seat in the US, you know how large, how tall, how wide, how heavy the individuals are, how long their arms are. You know, we don't know that. We don't know how dense the average finger is. We don't know what the range of densities are. We will and we continue to discuss what the optical characteristics of the fingers even look like. So until we can nail some of that stuff down, you can't say, well, your device should measure perfusion, which is an arbitrary number, down to the following.
Thank you. All right, a couple of last, I forget now, Jerry, did you have your hand up?
No.
Okay, I have a quick question that's related to what Boomy was talking about, and that's going to you, Carolyn. Help me to understand, what do you see as the barriers to enrollment when you're dealing with a freshly hospitalized patient?
Yeah, thanks for the opportunity to answer that question. And I'll start. And I also want to invite Kelvin Moore, who is one of our key study personnel. He's a medical student who really helped get us up and running at the general and has recruited in that population before medical school as well. But I'll take a first start. I have found patients to be incredibly generous with their time. They have heard about this problem in the media. They also understand that there are care problems that are different for people who have walked different paths through life, and they want to be solution oriented. They, by and large, say, yes, I would like to help. Like, please sign me up. They appreciate that we approach them with sensitivity and offer to come back when they don't, when they're feeling well enough to talk. Families have been generous. We use language concordance that helps tremendously. And an important part of this also is that we actually have a modified consent protocol. So some people call it a waiver of consent, where we can collect minimal risk data when someone is too unwell to participate in a conversation. We do come back and speak with them when they've recovered enough to talk. But I just want to untangle some of the hesitancy to approach patient populations. I think offering them participation in developing data that impacts them and people like them and their families and grandchildren is a moral imperative, and offering access to research participation advances healthcare for the people who have been totally underserved by this technology, so far as we can tell. So I want to just invite Kelvin to add any comments about working with hospitalized patients.
Sure. Thank you, Carolyn. Hi, everyone. Once again, Kelvin Moore, medical student, also working with the Equiox team here at Pulse Oximetry. I think in addition,
Kelvin, can you speak up a little bit? There's a little bit of audio issue.
Sure. In addition to the points that Carolyn has raised, I think what's really important, and Boomy also spoke to this, is patients seeing folks, or the recruiters looking like the patients that they're actually trying to recruit into the study. In my experience as working as a clinical research coordinator prior to med school at the General Hospital, it was 4 studies trying to recruit Black men who have sex with men. And in the past, the research team told me that they really had difficulty recruiting that population. And so when I would go to these affinity groups, I felt that there was more trust. And that sort of speaks to the power of representation in clinical research. Of course, there's a lot of barriers to, having the recruiters look like the people that they're trying to recruit. But I think that's a very powerful tool in terms of trying to diversify your research cohort or sample. And then additionally, Bunmi spoke to this, but the power of accessibility to research is very important. And so anecdotally for San Francisco, a lot of Black individuals who live here in the historically Black neighborhoods, it's very hard to get to downtown San Francisco by way of the bus routes. And so just thinking about structural racism on that front, it's hard to get these people to the hospitals, to the sites where we're actually doing the studies, because there is not the infrastructure, or the infrastructure rather, does not cater their experience or is not tailored to their experience. So I think in addition to all of the things that Carolyn has raised and Bunmi expertly raised, representation is very important in the patient population as well as on the research side. And then also thinking about the accessibility and transportation needs of patients is very important, because in the US and I'm sure internationally as well, there are a lot of different structural racism or policies or different transportation barriers that sort of preclude folks from underserved populations, from lower socioeconomic backgrounds, from actually reaching hospitals, not only for their healthcare, but also for research.
Thank you. So one of the challenges I know when doing this in a controlled laboratory setting is you have various exclusion criteria. If their carboxy level is at a level, and they lied about the fact that they're non-smokers, they're excluded because you have a metric where you can say, oh, you're not going to participate. That's not real world. The person in the ED that's anxious and had a stroke, they may have been smoking, 2 packs a day for 40 years. So is it appropriate to have that as an exclusion criteria for real world testing? Carolyn, do you do that?
No.
Okay.
And I would offer that I would be curious about what that patient's carboxyhemoglobin level was from. Like maybe they were standing at the bus stop for 2 hours waiting to come in. So they may or may not be just misrepresenting their tobacco use, but we don't exclude patients for anything besides protected research populations. So people who are pregnant or people who are in custody of law enforcement because they're subject to coercion. But other than that, we just try to collect the covariates that may become important in our analyses. And real world clinical research is super messy. And that is just the nature of the beast. And so you have to, at the planning stages, be open to all the questions you think you may want to ask, get those variables in your data set, and also enroll sufficiently large populations with enough spread in the data ranges that you really care about. And that's the part we're going to struggle with, is getting those sort of lower 90s oxygen saturations well represented.
Well,
Just to add one thing about it.
yes.
We are though collecting COAX data. We do have the carboxy levels on all the patients. Like we have that information, but it's not excluded.
That was my
We will
understanding.
be able to account for it.
Yeah, we don't exclude, we get more data.
Yep. That was my understanding as well. I just wanted to make sure it was clear, because a lot of people around the table have only lived with normal low carboxy met values because that was an exclusion criteria. So these are the realities of what is happening in the way of real patients. And when I think of a euphemism, perhaps, in my experience with multiple pulse oximeter designers and manufacturers, and as a clinician user, the term outlier has been there. And guess what? The outlier is a real human breathing subject and needing medical care. But oftentimes this outlier is in the controlled desaturation study. And I hear manufacturers saying, oh, they corrupted all our data. Well, I agree that this N of 1, when compared to 12 other subjects, may be a heavy change to the values, but it was real data. So you have to realize that these comments that you hear about cardiovascular function, et cetera, or maybe it's individual physiology with how they can tolerate hypoxia, is there. but realize normal variability in pulse oximeter values, it's part of life. If you don't have variability in your heart rate as well as in your saturation, you're decorticate. I mean, this is not normal. So variability is a good thing. Yes.
Yeah, so one of the things that I liked about Sandy's presentation is that he separated the engineering and the clinical.
Yes.
And that's important. When we do these studies, we have to ask a question, right? What's our hypothesis? Are we evaluating racial bias in the real world? Or are we evaluating carboxy hemoglobin influence in the real world? But so when we start to collect this data, the important question is always the hypothesis. What is it that we're trying to do? In the DSAT and the control DSAT study, what I view from what I've read in the past is we're trying to calibrate, or we're trying to verify the calibration of this pulse oximeter, right? We're trying to determine, did we include enough of the darker subjects? We include enough of the lighter subjects so that when we have a calibration curve, right, that it theoretically will fit into a validation model, right? So when I look at that study and I say, okay, if skin pigment is a factor, then I should consider the very dark pigment. I should consider a medium pigment. I should consider a light pigment, because in that sense, then I can have a sense of where I am in the spectrum of my calibration, right? When I go to the real world, right, the one thing that I would say is, what is the real condition, right? And one of the things that's come up, and I think Dr. Bickler pointed it out, I think several people have alluded to it, is the sensor placement, right? In the lab, people can be very controlled about how they place the sensor, right? I think a lot of people in this study have done desaturation studies, have done a very good job of understanding that sensor placement. What we learned today from some of the examples is that people aren't necessarily looking at the sensor placement, but they're looking at the number that's generated from the placement, right? So they're adjusting the number. This number looked too low at 80. If I put it on the ear, I get 90. Okay, that's a good number, right? And so one of the things that I think is helpful, and when we start to uncover, and what Elias Monk said about colorism in this segment is very important. I think we start to uncover those layers, and we say, is it colorism? Sure. Is it sensor placement? Is it a manufacturer calibration curve issue, right? And I think those things we need to start to layer by asking the right question for each study. We looked at the pulse oximetry data with a perfusion index, and we saw this delay. But did we ask ourselves, why is there the delay? Why is it that there is a difference? Did we look at the placement of that sensor? Did we just decide that we needed to replace the sensor? And so a lot of these things, and when we talked about Mark, he brought up this measure of uncertainty. And one of the biggest things of pulse oximetry is the detection mechanism. The detection mechanism is the largest barrier of getting a correct, accurate measurement. Anybody who's worked with a pulse oximeter knows you misplace it, you can get a wrong reading. You misplace it, you can get a reading that's not responsive. So everybody knows this from perspective, but when we do the real world clinical study, Michael Schroding showed a bias, right? But did we look at, were they using the right sensor? I saw a sensor picture where the sensor was on the thumb. Not many people have validated a thumb as a sensor location for an adult.
Blindness. I'm going to take a prerogative here because some of those subjects we're definitely going to be talking about more tomorrow.
Right.
And I think everyone is in agreement that the type of studies, many of which contain flaws. And I think one of our goals in that regard is to make sure that good education and available resources are there. And such resources that now Carolyn has put together, the Stanford group has done it for children, allow for those conditions as best as they can be done for the bedside clinical data gathering, not routine use. We're going to try, if we can show that refining what goes on no longer puts the data that the manufacturer claimed for their ARMS to be fourfold greater in real world use, that's unacceptable, right? And it's largely because people are careless. Carolyn is careful. And the same is true of the Stanford effort. And those protocols will be widely available. I look forward to seeing the data. The preliminary stuff looks exciting, that it can be very, very good.
I think
Now,
the one I would just emphasize, the point I was trying to make is we got to answer the right question. We got to ask the right question, and we got to make sure we're not blending studies, because one of the things that is getting confused now is we're talking about racial bias studies, but then we're conflating it with other factors, or we're conflating it with sensor placement issues. And so what I'm saying is let's make sure that we're controlling the variables we need to control so we can get the right answers that we want. Because honestly, there is, to me, I'm concerned, right, that we are masking colorism, right, because there may be a bias there that we're not accounting for in our pulse oximetry data. And that's something that, so one of the things that I've always kind of wondered is, why don't we take, and I think, what was it, one of Julian Goldman brought up, why don't we look at the continuous data to see where it was, right? And some of those aspects of it, because my concern is, are we masking, and honestly, pulse oximetry has done good. And a lot of you have worked in that field, and so it's done good. But at the same time, we need to do good for everyone. But are we being fair to I guess that segment of the population and saying, well, we're looking at it not holistic, we're looking at it more holistically than just pulse oximetry.
I'm going to take prerogative again here.
Sorry.
All right. So
You have two questions on
Yes.
all Bob, but we're not.
yeah, do you want to go for
Phil,
the,
could you call? I think go with maybe Dr. Conrad first. We've got about 5 minutes, and we'll need to move on.
okay, yeah, I want to make sure that we are okay. So there are several comments in there from Gorham. Gorham, it's very late for you. I appreciate you being up. It's now 11 o'clock. So thank you for your depth of questions there, but people can certainly look at those because they talk to real world conditions of low blood pressures with sepsis, etc. And please communicate with these individuals privately. Gorham is really an expert in this space, especially in baby care. All right. I will move beyond that one.
Dr. Conrad, can you hear us?
Yes.
You can go ahead and take yourself off mute and chime in.
Yeah, why don't you do that?
Well, I feel like now we've gone like way off course. I was just talking more about the recruitment and the, including equity in our populations and things like that. And just wondering also, we talk about differences in race, but also a big barrier too is language. And I know that one of the issues that we had brought up to our own research entity within Stanford and trying to promote overall increased research equity is eliminating the cost barrier of us having to pay in-person interpreters. So we do have that availability at our hospitals and things like that 24/7, but in the past they used to charge us to use them. And then that kind of also would limit people's ability to increase that. So something else to think about as people are doing real world studies as well, and other thoughts about that.
Yeah, certainly a good point. I guess I'm going to go back to Carolyn on making certain that she showed pictures of different translations that were part of her cart, her portable cart. That made, it's best efforts. And Linus, to the point there, is all comers is what is needed. We're not super careful about a hypothesis other than matching real world conditions of end users. And that's end users as clinicians as well as the human subjects. But anything else you've now learned that may improve access to enrollment?
Yeah, to Calvin's point, not only looking like the population you're trying to invite to participate, but sharing a language preference with that population. So when you're hiring and you know your patient population has a language preference, it's not represented on your team, that should be one of your core preferred qualities that you take to HR. Because having a direct conversation is always better than using interpreter. And we use medical interpreters for all of our consent conversations. Dr. Conrad, please don't tell ZSFGH, but I just don't tell them that I'm having a research conversation. I just call the interpreter and have a research conversation without paying differently. So I feel like they're saying they're advancing healthcare worldwide for everybody, and that includes facilitating research conversations. So I don't pay for the interpreters.
1000%. We've done the same until we kind of got them on board to see the importance of this, but that's just other considerations for other people as a barrier. But our in-person interpreters are amazing, and it's definitely a difference than when you're using the
Now I have to say, coming back to carboxy, the question of how do you test perfusion? I mean, as a clinician, you know, and you're limited in your resources. You look at capillary refill, but the first thing I will do, you know, it's not on. I still am part of a volunteer fire department out in our rural community. You know, you're not going to put a pulse oximeter on the smoking hand, okay? Because those fingers really don't look right. So you look at your site as well as making sure the sensor is appropriately placed. So Mike, I know we've gone just a little over. I kept saying it's okay. I hope it is.
Yeah, I think we're in good shape. We're gonna get ready to move on to the next session. Thanks very much for chairing that one, to the speakers and that panel. I think there's some discussion in the chat right now. Jim brought up a great point, and Ellis responded about, you know, how representative much of the testing is being done in the US, for example, or certain countries. What is the applicability to the rest of the world, arguably the majority of the world, which is not necessarily represented? I think let's come back to that tomorrow morning on the discussion of skin quantification techniques and the specifics of what diversity looks like for the purposes of these studies. I think that's a really important point. Thanks for bringing it up, Jim. And we'll make sure tomorrow Dr. Lester is going to be and Kelvin are going to be co-hosting that session, so we can make sure we discuss that with adequate time. So thanks for bringing that up.